Today we're going to turn all that sensory input from the visual system and the auditory system and the somatosensory system, etc., and turn that into motor output, right? So, from a basic mechanics, we are input-output organisms. Obviously, there's a lot more going on than just inputs and outputs, but that's what we're focusing on here. So we're going to talk about the motor system, muscle, and the nervous system that controls muscle. So we'll talk about the type of muscle. We're going to focus on skeletal muscle for this course, but in the other A&P course, cardiac and smooth muscle are a focus, but not here. We'll talk about the types of skeletal muscle, introduce some concepts of the motor unit and the special synapse at the neuromuscular junction, the neuromuscular synapse. Talk about the mechanisms of contraction of skeletal muscle. That's what we're going to do in the first part. And then we will we're going to focus on the output first, and then we will move upstream. Talk about motor neurons that control skeletal muscle. Talk about circuits in the spinal cord that control the motor neurons that control muscle. So there's a lot of processing actually in your spinal cord. It's not just a transmission line, it's also a part of your central nervous system. And then we'll talk about how the brain, basal nuclei, cerebellum, motor cortex, and subcortical regions, all of this inputs the lower structures that control muscle. Note, this material will be on the exam. So the exam, which is on Wednesday, includes sensory and motor systems. Just want to make that clear. I know the exam is soon, but you'll have 48 hours to process this information for the exam. The answers to the problem set are on Moodle now, and the answers to the anatomy quiz will be on Moodle by the end of the day today. In fact, they'll be by the end of the day today. Okay, so let's talk about the types of muscle. We're going to focus today on skeletal muscle. It's called skeletal muscle because it moves the skeleton mostly. There are some skeletal muscles that don't move bone, like the diaphragm doesn't move bone, but it is a skeletal muscle. But most of the muscles we're talking about move bone. Let's do some calisthenics. Everybody get up. Let's review some of the movements that you have learned about. So I would like you to abduct both of your arms. Keep your elbows extended, abduct, or let's get into the anatomically neutral position. Abduct both of your arms. That's abduction. Okay, let's go to the anatomically neutral position. Abduct one of your legs. Obviously you can't do them both at the same time. Okay, abduction of the leg. And then adduction of the leg, right? Abduction, adduction. Okay, let's flex your elbow. And then extend your elbow. You can't hyperextend your elbow without hurting yourself. Flex your shoulder. Flex your shoulder. Flex your arm at your shoulder, not your forearm. Right? So if I'm in the anatomically neutral position, that would be pure flexion of the shoulder. I could then do some pronation, but let's not do that. Flexion of the shoulder, extension of the shoulder. Okay, that's review and some new material. So let's talk about the movement of the hip and the knee, because that's what we're going to be doing in lab this week. And you have done in the coloring book. and could be on the exam, right? Let me just make that clear. So go ahead and flex one of your legs at the hip. Keep your knee extended and flex your leg at the hip. That's flexion of the leg at the hip. So go ahead and flex your leg at the hip and then flex your knee. Right? So this is flexion of the knee and extension of the knee. Flexion of the hip, extension of the hip. So I'm sorry, people online, you can't see my lower leg, but let me try to do that again where you can see what I'm talking about. Okay. Flexion of the hip, flexion of the knee. Extension of the hip with the knee extended, and now with the knee flexed. Okay? Let's do plantar flexion of the foot. Plantar flexion of the foot. Right? Plantar flexion. So now I can do plantar flexion of both feet at the same time by standing on my tippy toes. Dorsiflexion would be standing on my heels. So dorsiflexion, plantar flexion. Really, dorsiflexion is extension, and plantar flexion is flexion. I don't know why they screwed it up like that. Some of you may know the name of the surface of the bottom of the foot is the plantar surface. Some of you have had plantar fasciitis or some sort of kind of pain in your foot related to inflammation in the plantar surface, or plantar warts, which are warts on the bottom of your feet that get quite painful. So the way I remember it is plantar flexion is bending the foot towards the plantar surface. That's plantar flexion. Dorsiflexion is standing on your heels. So some movement of the lower limb from the coloring book that we'll do in lab, but could be a little bit on the exam, just to keep you honest about your work in the coloring book. Okay? Any questions about movements? Abduction, adduction, flexion, extension, flexion, extension, plantar flexion, dorsiflexion. And knowing which muscles do which. You can kind of feel it. You can feel when I do this, I can feel the muscles in my calves, my gastrocnemius and my soleus. When I do this, I can feel the tibialis anterior and the extensor digitorum longus working. So let's go ahead and get started on muscle now that we've used some muscle. So we're focusing on skeletal muscle. Skeletal muscle is muscle that typically moves bone. Muscle is connected to bone by tendons. Muscle ain't like the biceps brachii, or the biceps femoris. The muscles are made up of muscle fibers. Muscle fibers are the cells, the adult cells of the muscle. Muscle fibers are made up of, in embryological development, muscle fibers are big, long cells. I mean, they can be inches long, feet long. Big, long cells that are formed by the fusion of, individual cellular cells during embryogenesis. So there are stem cells called myoblasts that fuse during development
to make these big, long muscle cells, muscle fibers. Muscle fibers have many nuclei because they are formed from many individual cells, each of which has a nucleus. Muscle fibers have many nuclei, as I just said. The working end of a muscle cell is called the myofibril. It's not the end. The working structure within the muscle cell is called the myofibril. So we have muscles, muscle fibers, and myofibrils. Myofibrils are microscopic. Muscle fibers are microscopic, although so microscopic in diameter, inches or feet long in length. We'll talk about the structure of myofibrils in a few more slides. There's some anatomy names here that I do want you to know. It's introduced here, but we'll talk about it more in a few slides. So a skeletal muscle is connected to bone via a tendon. Some of the tendons that we have seen in the lab are very clear and beautiful in some ways. Some of them are not so clear, but they're all there. They're there one way or the other. They can be short and not easy to see. They can be very long, like the palmaris longus tendon is inches long. A muscle is surrounded by an epimysium. That's the shiny surface on a muscle. And it specializes at the end to form the tendon. So the tendon is a specialization of the epimysium. Within the muscle, there are bundles of muscle fibers. So each of these pink dots is supposed to be a muscle fiber cut in cross-section. So there are bundles of muscle fibers called fascicles, each of them wrapped by a structure, a membrane structure called the perimysium. And then each muscle fiber within the fascicle is wrapped by an extracellular matrix called the endomysium. And the reason we have all this matrix, extracellular matrix here, a membrane extracellular matrix here called the perimysium, extracellular matrix here called the epimysium, it gives strength to the muscle. After all, the muscle is moving things, lifting weight, generating force, a lot of force in some cases, and cells are not particularly strong, cell membranes are not strong. So in order to prevent rupture of muscles, they are supported by extensive systems of endomysium, perimysium, and epimysium to give them mechanical support and strength. If you tear a muscle, you're tearing all of, if you injure a muscle and you tear a muscle, you're injuring all of that. And that's, and then you're also injuring blood vessels. And that's what leads to a bruise, or that's what leads to pain and, healing if you extended a muscle beyond its abilities. There are three types of skeletal muscle. So we're not talking about cardiac and smooth anymore. We're talking about skeletal. And within skeletal, there are three types. And let's just learn about them. So there's the slow twitch muscle, which its name would suggest is slow. These are postural muscles. These are muscles that are working all the time, or almost all the time, to maintain posture. So right now there are muscles in my back that are contracting, working to keep my back erect. There are muscles in my hips that are working to keep my legs, my hips, balanced and keep me upright. So these are muscles that are working all the time. So they have lots of mitochondria for the production of lots of ATP by oxidative phosphorylation. And they have lots of a protein called myoglobin, which is an oxygen. It's like hemoglobin in your blood, except that it's not hemoglobin, it's myoglobin. but it's an oxygen binding protein. So it's there to sort of store oxygen so that, if you hold your breath for a minute, this muscle can still have the oxygen it needs to do oxidative production, oxidative phosphorylation production of lots of ATP. So it never runs out of ATP as long as it's getting oxygen and blood. It expresses a slow form of myosin. You learned about myosin in cell-phys. We're going to build on that now. Myosin, the type of myosin in skeletal muscle, has several isoforms, and the isoform that is expressed in slow twitch muscle, you'll see in a minute why we call it twitch. Slow twitch muscle is a slow myosin. So it's not specialized for rapid contractions, it's specialized for continuous contractions. It's red in color. So let's jump to the fatigable fast twitch muscles. These are the muscles that are specialized for fastest contraction. They have the fastest isoform of myosin. They produce their ATP by glycolysis. You learned about glycolysis in cell-phys. Remember, glycolysis is some of the input steps to, input to the Krebs cycle, and which then inputs to oxidative phosphorylation. It's not very efficient. It's not efficient at all, but it is fast. So this produces ATP in fast bursts. But it runs out of ATP very quickly because it's inefficient. And so it's fatigable. This is a muscle that would run out of ATP fast. ATP is produced by glycolysis, fast but not efficient, runs out of ATP. It doesn't have a storage protein for oxygen. And so some of your muscles that are fast twitch are the muscles of your eye. They don't have to work, they're not working to hold the position of the eye. They're specialized for moving the eye very quickly, or for blinking. Your eyelids are not very heavy, so you can blink your eyes very quickly. But if you tried to lift a weight with your eyelid, it would become fatigued very quickly. If you were a bird, your flight muscles would be white because they have to contract very quickly in order to generate the force for flight. If you're a chicken, these are leg muscles, dark meat, and this is flight muscles, the breast muscles needed for flight, the pectoralis major muscles of the chicken, or even a flying bird, are white meat. In between are the fatigue-resistant fast twitch. So these are identifiably different from fatigable fast twitch and from slow twitch. They are fast because they have an intermediate speed myosin. They produce ATP by oxidative phosphorylation and glycolysis. So they can maintain ATP, so they are fatigue resistant. But they're fast. If you want to learn the names type 1, type 2A, and 2B, that's fine with me. But I think the descriptive names are more useful. Slow, fatigue-resistant fast, fatigable fast. Most of your muscles are a mixture of fatigable fast, white, no myoglobin, no mitochondria. Slow, red, lots of myoglobin, lots of mitochondria. And pink-ish. Fatigue resistant, but fast. So this is a good example of the three types of muscle cells, muscle fibers, in each muscle, in a muscle. Most of your muscles are a mixture. Some of them are more, like my gluteus maximus, which is working now to keep me standing upright, or my erector spiny muscles in my back. They're mostly slow fibers, but not exclusive. My eyelids are mostly fast muscle, but not exclusive. Maybe they may be exclusive, I'm really not sure. And then my biceps are a mixture, because my biceps can maintain posture. If I were to hold my arm here, I could hold it like this for a long time, even if I was holding a moderate amount of weight. But it's also meant for, generating force quickly if I need to. Questions about the types of muscle? Each muscle fiber, each muscle cell, remember a muscle cell and a muscle fiber are the same thing, is innervated by a motor neuron. So here is the motor end plate. It's got a name. It's the neuromuscular synapse. So here is the synapse on this cell. And here's the synapse on this cell, and this cell, and this cell. Each muscle fiber receives innervation from one motor neuron, and there's one synapse. So this synapse, which tells this cell to contract, has to do so by an action potential. We're going to review this in a minute. Action potential, neurotransmitter release, excitation, and then an action potential spreading on the surface of the muscle. Muscles fire action potentials in order to spread the excitation from the one synapse, all the way from one end of the cell to the other. So this is a specialized synapse, which we'll talk about in a minute. So I said that each muscle fiber is innervated by one motor neuron, but each motor neuron may innervate many muscle fibers. So so here is a motor neuron. Well, let's take this one, because this is a bundle of motor neurons. Here is one motor neuron that splits here and innervates these two cells, and maybe some others that are off the screen. Here is one. Let's just say it splits here and it innervates this one and this one. So it's hard to see, but in cartoon, one motor neuron can innervate several muscle fibers, but each muscle fiber receives innervation from only one motor neuron. The set of muscle fibers that is innervated by a single motor neuron has a name. It's called the motor unit. The muscle fibers innervated by one motor neuron is the motor unit. So here's a cartoon of two motor units. The purple, is colored to help you see. The purple motor neuron is innervating a set of muscle fibers in a muscle. Another motor neuron is innervating another set of muscle fibers in a muscle. The control of a muscle might have hundreds of motor neurons controlling hundreds of motor units in a single muscle. So some muscles have many motor units, and some muscles have only a few motor units. I don't think there's any muscle that is made up of one motor unit, that is innervated by one motor neuron. I think all muscles are innervated by several motor neurons. That means they have several motor units. Question about this definition of motor neuron, the synapse, branching, and the definition of a motor unit. All right, then let's focus on the neuromuscular synapse. It's a special, you learned about synapses. We're going to reinforce some of your knowledge about synapses, but this is a special one. It's not a neuron to a neuron. It's not a presynaptic neuron interacting with a postsynaptic neuron. In this case, the postsynaptic cell is a muscle. So it's the neuromuscular junction. So let's get oriented here. Here's the neuron. How do I know it's the neuron? It's filled with vesicles. The vesicles are packed with neurotransmitter. And at the neuromuscular synapse, the neurotransmitter is acetylcholine. So you've learned in some cases that I do want you to know the name of this neurotransmitter at a synapse, like the name of the neurotransmitter that is at the synapse between the retinal photoreceptor and the bipolar cell has a name. You know the name of that neurotransmitter. I'm asking you to know that one. I'm asking you to know this one too. That the neurotransmitter at the neuromuscular synapse is acetylcholine. So here's the membrane of the nerve. And here is the membrane of the muscle underneath the synapse. Again, let's go back to the image. So here is the nerve cell. Here is the presynaptic terminal of the nerve. And then there's a membrane of the muscle. Nerve, membrane of the nerve, membrane of the muscle underneath at the synapse. There's a specialization of the muscle cell with these folds underneath the neuron at the synapse. So these are junctional folds. Junction because this is the neuromuscular junction. And the receptors for acetylcholine are highly expressed at the tops of these folds. So this area right here and this area right here are where the receptors are highly expressed, and the receptor is a nicotinic acetylcholine receptor. You worked with this receptor before, all the way back to problem set 1. This is an ionotropic receptor that is permeable to sodium and potassium. And because it's permeable to sodium, when it is open, sodium flows in and depolarizes this muscle cell. So the next slide on your handout has a blank where there should be information. In keeping with our pattern, I'm going to assign this as homework. You should be able to fill in the blank. Let me just, the first sentence is, an action potential propagates in the neuron. Well, I don't know, let's spend 5 minutes here doing this one together. Take a little time. An action potential propagates into the neuron. Propagates along the axon into the presynaptic terminal of the motor neuron. Okay, what's the next step? Now, you're off and running. You know everything you need to know because it's the same story that you have learned with just a couple of different twists. What's the next step that happens after the action potential propagates into the presynaptic terminal? Anybody, online students too, we can hear you, so go ahead and chime in. Speak louder. Voltage-gated calcium channels are opened by that depolarization. Good. What's next? Calcium influx into the presynaptic terminal. I could ask you what maintained calcium low in the presynaptic terminal. Well, we've got those calcium ATPases, CERCA, PMCA, and NCX. So that's going all the way. I'm just using this to reinforce information that I hope you haven't completely forgotten because we're using it now. Okay, calcium goes up in the cytoplasm of the presynaptic terminal. To what does calcium bind? You know the answer. Synaptotagmin. When calcium binds to synaptotagmin and causes the conformational change there, what does it cause the conformational change of? What's the name of the protein that is going to be important next in the next part of the story? They're complexes. They've got initials, both of which start with, yeah. Say again? B snares and T snares. The reason I can't hear you is this thing is humming over here, and I've lost my high-pitched sound in my auditory system. Anyway, B snares and T snares, conformational change, fusion of the vesicle. What's the name of the neurotransmitter that is released into the synapse between the nerve and the muscle? Acetylcholine. What does the blue illustrate? The location of the receptors, the nicotinic acetylcholine receptors. So acetylcholine diffuses, binds, activates nicotinic receptors. What happens then? What's next? Single word, begins with a D. Depolarization. Sodium influx through nicotinic receptors, depolarization of the muscle cell. We've just moved from excitation of the neuron to excitation of the muscle. Neuromuscular excitation. Depolarization of the membrane underneath the synapse. The green illustrates the expression of voltage-gated sodium channels, and it extends all the way out on the muscle. So from here to the end of the building, from here to Tyler. Voltage-gated sodium channels. What did we just do? This depolarization opens voltage-gated sodium channels because we reach threshold, and the muscle fires an action potential. So I think that's where we end this story, because we have not yet explained how we're going to get contraction of the muscle. This is a cartoon of the contractile machinery. So here's the answer, which I will post. I think we got it all. Action potential propagation. I didn't use the word here, but action potentials propagate the entire length of the muscle. Same reason that action potentials propagate on unmyelinated nerves. Questions about this? So this is key concept from a while ago that we are using again. Now, we're going to talk about calcium handling in the muscle cell, because calcium is going to be the trigger for contraction. So here's a cartoon of a muscle cell. Surface membrane, sarcoplasmic reticulum, contractile machinery. Sarcoplasmic reticulum. What was the protein that pumped calcium into the sarcoplasmic reticulum when the muscle was relaxed? Circa. Same old, same old. Circa pumped calcium into the sarcoplasmic reticulum. And you can see in this cartoon, it's drawn as a reticulum, not just a sac of membrane, which is closer to the truth. And if you're interested in seeing this in a three-dimensional model, during the break you can come up and see it on this, greatly enlarged muscle fiber, which, shows it in 3 dimensions. Sarcoplasmic reticulum, high calcium in the lumen of the sarcoplasmic reticulum, low calcium in the cytoplasm because of circa. The action potential that spread along the surface of the membrane, surface of the cell along the surface membrane, propagates into these special structures called T-tubules. In the model here, they're this blue network. So there's a network of deep impressions of the surface membrane into the muscle. And so the action potential, this is contiguous with this, and this expresses voltage-gated sodium channels. So action potentials move into these T2s. All of them. Not just this one. Okay, now it gets tricky. Now in the T tubule, this is a blow-up of the T tubule and its junction with the SR, and this structure is called the triac. Some books will use that word. I don't use it very much. This junction between the membrane of the T tubule and the membrane of the SR is blown up here. So here's the membrane of the T tubule, and here's the membrane of the SR. In the membrane of the T tubule, in addition to voltage-gated sodium channels, there is a protein called I call it the voltage sensor. Your book calls it the dihydropyridine receptor. The voltage sensor is a physiological name, because that's what it does. It senses the voltage change by the action potential. DHP receptor is referring to the fact that it binds a drug called DHP. So this is the pharmacological name, and the physiological name is voltage sensor. And you can use either one. I will accept both. I will never require you to give me both, but you can use one or the other. So this protein is a voltage sensor. It senses the voltage, it senses the resting membrane potential, and it senses the depolarization by the action potential. When there is an action potential in here, it senses it and undergoes a conformational change, of course. The conformational change of this sensor protein is transmitted mechanically to the calcium release, to a calcium release channel that is expressed on the surface membrane of the SR, the SR membrane. And it opens those calcium release channels. I call them calcium release channels because that's a physiological name. That's not just me. They're called calcium release channels. Your book uses a pharmacological name called the ryanodine receptor. Again, either one is okay with me. So conformational change of the voltage sensor transmitted directly to the calcium release channel causes them to open, and calcium exits the SR into the cytoplasm. That calcium released from the SR is what's going to trigger contraction. And then what happens in relaxation? In relaxation, everything stops. Circa pumps calcium back into the SR, and the muscle stops contracting. There's Circa. There's the story of voltage-dependent calcium release. You've already seen some calcium release channels. They were the IP3 receptors in endoplasmic reticulum that responded to IP3 from GQ. So you've seen some calcium release channels from the SR or ER. These are related, but are different. These are not sensitive to IP3. They are sensitive to this mechanical coupling. Questions about the mechanism by which calcium is released from the SR when an action potential spreads along the surface of the muscle cell. Okay, now we're going to get into how the calcium controls contraction. How, when I contract my biceps, there is a calcium-dependent process to allow me to contract my biceps muscle. Let's introduce the players. The players, one player are the thin filaments that are made up of actin, troponin, and tropomyosin. These make up, here's an assembled thin filament. Actin, polymer, tropomyosin wrapping around it, sort of holding it together, and a troponin. There are different subunits of troponin that I'm not going to ask you to memorize. Troponin. Troponin, tropomyosin, and actin are the three main components of the thin filaments. And in a cartoon, a thin filament is cartooned here. And in a cartoon, a thin filament is cartooned here as the thin line. The proteins of the thin filament. The other protein complex necessary for contraction are called the thick filaments, and they are made up of bundles, many hundreds of individual monomers of myosin. So the myosin, the thick filament, are bundles of hundreds, maybe thousands, of myosin molecules. And they are bundled in a special way with the middle line. If I'm in the middle of a myosin thick filament, a bundle, then I have some monomers of myosin reaching out this way with their head, and I have some bundles of myosin reaching out this way. Right? So sort of mirror symmetric around the middle. Heads reaching out, myosin heads reaching out in both directions. So now let's build this into contraction. You've had some of this in cell, I think you've had most of this in cell phys. This is reinforcement from cell physiology. Here is a head group from a myosin, and here is it. So this is the thick filament on green above. One myosin head from hundreds that make up the bundle of the thick filament. Here's the actin, tropomyosin, troponin. They're very close to each other. The heads of a myosin form this sort of meshwork. The heads of the myosin are making contact with the thin filament. Molecular contact, molecular dimensions. Head, thin filament. When the muscle is relaxed, troponin does not have calcium bound to it because calcium concentrations are low. And troponin and tropomyosin block the site on actin that would bind, that wants to bind, that has affinity for the myosin head. So in low concentrations of calcium, muscle relaxed, can be stretched, troponin and tropomyosin block the binding site for myosin of actin. Competitive inhibition. In the presence of calcium, where did the calcium come from? From the SR. Why did that get released? An action potential, just to reinforce. When the calcium in the cytoplasm goes up, it binds to troponin. So the calcium binding protein here is not synaptotagmin, it's troponin. The calcium binding protein of the muscle is troponin. When calcium binds to troponin, conformational change moves the tropomyosin and the troponin away. Actin, now the binding site for myosin is now exposed. Actin can bind the myosin head group, and we're going to go off into a cycle of phosphorylation and conformational changes that are going to shorten the muscle length. So calcium triggers contraction by binding to troponin and moving tropomyosin and troponin out of the way so that myosin can interact with actin, so that thick filament can interact with the thin filament. At this point, we have contraction going on. So here's a cartoon from your textbook that illustrates contraction. The thin filament is connected to part of the muscle called the Z line. So here's a thin filament. Let's see if I can pull up let me look at the next slide. So here is the Z line in cartoon, and the thin filaments sticking out from it. Okay, there's a Z line, thin filament sticking out from it, in both directions. Here is the other Z line, thin filament sticking out of it. Thin filaments are in purple, blue, Z lines are the zigzag. Here's a real micrograph, here's the cartoon. When the muscle, in the absence of calcium, the myosin cannot interact with its thin filament because troponin and tropomyosin are blocked. Calcium, now we stimulate the muscle. Intracellular calcium rises because it's released from the SR. Now the myosin head can interact with the actin of the thin filament because the troponin and tropomyosin have gotten out of the way. This binding causes the ADP and phosphate that were bound to myosin, and you'll see where that comes from in a minute when we finish the cycle. ADP and phosphate were bound, they unbind, and that causes a conformational change from the extended myosin conformation to the flexed myosin conformation. And since this is attached to this, it pulls the Z disc in this direction. And so these myosin heads bind to this thin filament and pull it in this direction. These myosin heads interact with this thin filament and pull it in this direction. And so you can see that these Z discs get closer together. And that's contraction of the skeletal muscle. This anti-parallel structure pulling this way, shortening the skeletal muscle. So let's go back. So that was just one tiny little piece of it. The point is ADP and phosphate dissociation triggers this movement like this, which is called the power stroke. This movement here, number 2, is the power stroke. You should label your slide. Number 2 is the power stroke. It's caused by ADP and phosphate dissociation from myosin. All right, we got to make a cycle, so we're not there yet. What happens now? ATP binds. So as long as this muscle is healthy, it has mitochondria and glycolytic enzymes to make ATP. Whether it's fast twitch or slow twitch, it has plenty of ATP. If it's healthy, ATP binds to myosin because the ADP and phosphate unbound, leaving a site available for ATP to bind. ATP binding causes the myosin to let go of the actin. So even though the site is available, because tropomyosin has moved troponin has moved tropomyosin out of the way, when the myosin is bound to ATP, it has a low affinity for those sites. But myosin is an ATP ace. Under these conditions, myosin hydrolyzes ATP. The hydrolysis of ATP does two things. It puts it in the ADP bound state that has high affinity for troponin, for actin, and it causes the myosin heads, which were like this. The ATP hydrolysis causes the myosin heads to extend. So dissociation of ADP and phosphate does the power stroke, and ATP hydrolysis reaches out again. And so now you can see, if we do this hundreds of times, and it's not synchronized, so this, this bunch of heads might be, this bunch of heads might be attached, while this bunch of heads are relaxed, and this, detached. Anyway, it's not synchronous. These heads are wiggling back and forth, pulling the Z disk towards the middle, shortening this structure, and you do it here, and you do it here, and you do it here, and you do it there and there and there, and the muscle gets shorter. And that's contraction of the skeletal muscle. Now let's introduce the names that I do want you to know. So we've introduced actin, troponin, and tropomyosin. Myosin, which makes the thick filament. The M line is the name of the middle of the thick filament. The Z line is the name of the end, is where the thin filaments are attached. From Z line to Z line is called the sarcomere. So the sarcomeres are what's getting shorter. Each sarcomere gets a fraction of a millimeter shorter. Got 1,000 of them lined up. The muscle cell goes from, let's say, 4 inches long to 3 inches long. Z disc, M line. Actin, thin filament. Myosin, thick filament. Around the M line is a zone called the H zone. Around the Z line is a zone called the I band. So the I band is around the Z. The H zone is around the M. And the A band is where myosin and actin overlap. So from here to here is the A band. This is part of the anatomy of a skeletal muscle. And here's a cartoon that shows the contraction. So here is the relaxed muscle, and here is the contracted muscle. What has gotten shorter? Length of myosin? No. The length of the myosin thick filament has not gotten shorter, so the A band has not gotten shorter. The thin filaments have not gotten shorter, but what has gotten shorter is the region at which there was no overlap, because this muscle, these thick filaments have pulled this Z disc this way. So the I band gets shorter. The I band gets shorter. The H zone also gets shorter for the same reason. These have been pulled this way and shortens the H zone. So parts, but not all, of the muscle fiber of the sarcomere gets shorter. I think if you know the names and you know the story, you can figure out which gets shorter and which doesn't get shorter. And I might ask you to do that. So the next slide, this one I will do as homework. So I'm going to ask you, as in homework, to put this in the order. This is a summary. This is just a summary of what is the mechanism of contraction of the sarcomere. The cross-bridge cycle. This cycle. This cycle, which shortens the sarcomeres. Asking you to use words to put them in order. Okay, so I'm gonna go real quick through the next slide because it's got the answer. But do this as homework. Put the cycle in order. And since it's a cycle, you can start anywhere, right? You can start anywhere. Start with any letter and then run the cycle. I'm just thinking about stopping here. Taking a break. Sure, why not? Let's go ahead and let's take a 10-minute break here. And then we'll come back and talk about control of skeletal muscle contraction, both by the muscle and by the nervous system. Okay, so let's take a quick break.